en unos segundos ya.
Ariana, I see you're online. Do you know if it's okay if they record this seminar?
Hi, can you listen?
Mm-hmm.
Okay, yeah. I was on mute, sorry. Yes, they can record the seminar, no problem. Okay, good afternoon to you, to all.
Thank you a lot, Ariana. Thank you. So, Omar, por favor, entonces, si me apoyas con esto y pues prácticamente estamos iniciando ya. Thank you a lot. Again, Stephen.
Hi.
¿Podemos empezar, doctor?
Okay.
Adelante, por favor. Gracias. Adelante.
So, Steve, we can start.
Yes. Good morning, everyone. It is a pleasure for me to introduce you to Dr. Stephen Rumbelow. He is in charge of the life science research of Croda, and he has been in this position since early 2020. His responsibilities are developing partnerships and collaborations between R&D teams and external organizations, both in academia and also in research institutes and customers. Prior to that, he developed and managed the US healthcare R&D team for several years, generating new high purity excipients and supporting performance claims data, specifically for the pharmaceutical and animal care industries. This includes products for all major delivery routes and applications, ranging from emulsifiers and dispersants through solvents and permeation enhancers. The team also generates accompanying application and claims data with an emphasis on the impact of excipient purity on drug delivery and performance. So, Stephen, I give you the word, and I hope everyone enjoys your seminar.
Before I start, may I just introduce Ariana Mireles from Croda, Brazil, who is the Regional Development Manager for Vaccines.
No, pero luego va a haber alguien más. Ella solo está introduciendo, ¿no?
Sí, hay alguien con el micrófono encendido. Yo introduje al doctor Stephen y él va a dar el seminario.
Sí, por favor apaguen sus micrófonos todos. Gracias.
Do you want to say a few words, Ariana, or are you okay?
Yeah, so good morning all. I started by saying good afternoon because it's afternoon in Brazil. So I am, as Steve said, I am the Sales Development Manager for Prescription Drugs and Human Vaccines for Latin America. I've been working for Croda for 7 years now, and it's a pleasure to have this opportunity to participate today and to present to you some technologies that we have to offer to the bioprocessing and biotherapeutic market. Thank you.
And just as a side note, I'll introduce our local representative from the company, Daniela Carrera, who is the person you'd need to reach out to if you need any samples or want to, speak to me regarding technical assistance. So Daniela, thank you for joining too. And now I'll start the seminar. This is really around about polysorbates and related surfactants and bioprocessing and biotherapeutic formulations. The thing to note here is that Croda has got a wide range of products into the pharmaceutical industry. It not only supplies high purity pharmaceutical excipients, we supply vaccine adjuvants, largely through the acquisition of our company in Denmark. So we supply aluminum phosphates and aluminum hydroxide based adjuvants, as well as saponin based ones as well. And we have an active vaccine development program with our colleagues in Avanti. Having mentioned Avanti, I should mention that we've also acquired recently Avanti lipids. And so we're very active in lipid mediated delivery, particularly with the current COVID vaccines, such as those made by Pfizer. We service a wide range of industries within the healthcare sector, ranging from, as I say, pharmaceutical type formulations for prescription drugs, consumer health, veterinary, and also we have our own APIs based on the omega-based fatty acids. So the purpose of this meeting today is really give an overview of the biopharma production, talk about some of the key aspects that our materials play in this process. These include cell culture, viral inactivation, purification, final fill. And then the second half of the talk, I'm going to talk about polysorbates in biopharma in general. So if you look at the overview of biopharma production, you have the bioreactor and we have the various stages going through this process. And we're going to talk about some particular key areas that surfactants play a key role in. So just going through the various sections. But here's the next slide, really the featured aspect. So cell culture on scale up. So really about the role of high purity poloxamers, about increasing and promoting cell viability, and also poloxamer products in general, and how they play a role in bioprocessing. We'll talk a little bit about viral inactivation. And here we're talking about the replacement of Triton X100, typically using things like superrefined polysorbate 80 and other chemistries. Just for you to note, Triton X100 is not REACH registered over in Europe, and customers are actually moving away from this material to look at other alternatives for viral inactivation in bioprocessing. Talk a little bit about protein A chromatography, and then also the role of superrefined benzyl alcohol for making custom PAB solutions. And then finally, we talk a little bit about final fill finish, and then before moving on to, as I say, the biopharmaceutical formulations aspect of this talk. So in cell culture, the main concern is cell viability and maintaining it. And then problems become more prominent with scale up. So we're well aware of the various things that happen and processes that happen during cell culture. These include agitation, turbulence, and this is all down to reactor design. They're all process related issues, but they can lead to a number of problems, particularly regarding cell viability. In addition, you can have effects happening like cell adherence to the reactor wall. And with that bearing in mind, we've got to look at the various materials used in the various reagent media and look at and take into account the potential toxicity of various impurities in them, which can impact upon things like yield and cell viability. There's a range of products out there that are currently used in bioprocessing in the sort of cell culture stage. These include things like cell cushioning to prevent shear stress. There's materials like Simpronic PEF 68, or also known as Poloxamer 188, We have anti-foaming agents to maintain the gas balance. Again, a range of symperonics. And then also, finally, we have surfactants that can be used to prevent adhesion, which prevents materials sticking to the actual reactor wall and being more readily available to the media. And these include things like superrefined polysorbates or indeed high purity grade polysorbates. Excuse me. So Polysorbate 188 and the bioprocessing challenges that it faces. There are a number of interfacial and colloidal interactions that take part in a bioreactor process, and that can impede upstream bioprocessing if not adequately controlled. Agitation and gas entrance velocity in cell suspensions can result in cell damage via a number of processes. They can include bubble bursting, shown here, foam entrapment, and impeller sparge or turbulence. Look at things like the bubble formation during oxygen sparging of a cell suspension in a bioreactor environment. You can actually see a loss in cell viability as the cells, typically Chinese hamster ovary cells, aggregate at the surface, and then as the bubble bursts in the foam, you can actually result in disruption of the cell wall and loss of viability. And there's actual publications showing over a period of time how the cells are actually lost during bioprocessing because of this kind of issue. Over the years, people have looked at multiple polymers to seeing whether they can be acted as shear protectants in cell cultures during bioprocessing. There are a number of requirements, but the main one is really to sort of maintain some surface activity with the ability to absorb both gas and cell interfaces. And the two mechanisms that surfactants actually protect the cells is actually decreasing the cell absorption into the bubbles themselves and actually strengthening the cell membrane to actually increase physical robustness. Of the range of materials that we've looked at polyxomers, polyvinyl alcohols, methylcelluloses, PEGs, maltopyranosides, and PVP, polyvinyl pyrrolidone. All these materials have generally been looked at in the past, but the one that is typically the industry standard is polyxomer, in particular Polysorbate 188. This is by far, as I say, the industry standard used in bioprocessing. Typically, it's used at fairly low concentrations, about 0.1% weight volume. Polyxomers are actually synthesized. It's fairly straightforward. You take polypropylene oxide, and then you polymerize that to form an actual core, and then you react that further with ethylene oxide to give you your copolymer. And the actual nomenclature of the polyxomer is actually a reflection of the ratio of EO and PO units in the product itself. And essentially, it consists of a non-ionic tri-block polymer with a central hydrophobic and two hydrophilic chains at either end. Well, Polysorbate 188 absorbs the cell membranes and does offer protection from shear stresses. But we have seen, and it's well known within the industry, that high molecular weight species present in the suspension can result in cell damage and actually decrease overall cell viability. And this has been a problem within the industry for a number of years. It's actually controlling the amount of high molecular weight species you might get present in a Polysorbate 188. So there's some results here showing basically you've got the surfactant acting as protecting agent, but here's one with a cartoon here with a surfactant with some surface active impurity. And you do get some cellular disruption arising from this. And if you look at the actual graph shown here, you've got two different concentrations of Polysorbate 188 and then a known added impurity, which is Polysorbate 407, has actually been spiked into this just to show the impact of what a high molecular weight species can do in terms of cell viability. And you can see that certainly at the sort of concentrations of even as low as 0.01%, you're getting quite a significant drop in viability. Obviously, this change is less at even lower concentrations, but still. So one of the things you can do is you can determine this high molecular weight species by size exclusion chromatography. So this is an example of what's been done in the industry and is fairly well known. So you can analyze for it by, as I say, size exclusion chromatography. And this is a technique that we use routinely when we've been developing high purity poloxamol 188 for bioprocessing. We have also developed an HPLC analysis to identify hydrophobic subspecies under that high molecular weight peak in poloxamol 188, because until fairly recently, it wasn't fully appreciated. It wasn't just high molecular weight species that were giving the problem with cell viability, but it was actually a hydrophobic subspecies in that group that was causing the problem. So we've actually based our in-house methods based on a rapid HPLC-based assay, which we actually recently published by Biogen. We've actually using this analysis to actually correlate the presence of the hydrophobic species in multiple lots of poloxamol 188 with cell viability in a shake flask CHO cell culture. So if you look at this graph here, you've got the high molecular weight species, and the hydrophobic species is actually this group here. So these are the higher molecular weight species coming off the HPLC column. But it's this subgroup here that's of interest to us. And again, as I say, Biogen, a US company, actually have correlated this hydrophobic species with cell viability. You see the viability drop can be correlated with the level of impurity present in this material. So one of the things we've done is to develop our own high purity product using our own in-house purification processes and also careful control of the reaction conditions used to make this polymer. And we've actually optimized and refined the process to eliminate both the high molecular weight and hydrophobic impurities with these two analyses, so both size exclusion and with HPLC. We then back this up by using a small scale shake flask based method for determining and confirm the impact of poloxamol 188 on purity on cell viability. So just to show you the typical sort of data we've been getting, we've looked at some commercial P188 material, and the aim is to produce material that's equivalent to, if not slightly better than. And so far we've been able to make 3 batches on a sort of semi scaled up process, and we're fairly confident, obviously, that we're going to be launching this material very soon, and it will be available in the new year. One other area we'd like to talk about is Viron Activation, and this is a technique that's being used fairly widely within the bioprocessing industry. And typically it's to do with envelope viruses and disrupting the outer membrane using surfactants. Typically what people use is Triton X100. Now, Triton 1X100 is still being used, but it's actually gradually being phased out. And I think as of next year will not be allowed to be used in certain countries, particularly Europe, and I think that's going to be spreading around the world as we move on. It's not regarded as, it's got degradation impurities that are of concern, basically. So Croda has its own products that can be used in this area. And I mean, one of the areas that people have looked at in particular is Polysorbate 80. Particularly when used in conjunction with triton-butylphosphate. There are actually numerous papers listing the use of tributylphosphate and Polysorbate 80 as a viable alternative to for antiviral use in the manufacture of human serum products already. So it's pretty well known within the industry you can use this combination of surfactants. And likewise, there's also publications coming out from the biotherapeutic field as well. There are needs, obviously, other materials have been considered, and there are sort of draft patents submitted for various non-ionic surfactants, such as bridges and merges, which are essentially, again, materials that could be used, but as I say, the most likely contender is to be Polysorbate 80. If you look at one case study, this was some recently published work by Chen et al. Looking at the viral inactivation of a couple of, well, Chinese hamster ovary cells, but in conjunction with a couple of viruses that they looked at, porcine parvovirus and pseudorabies virus. And they're looking at the the log reduction factor. And typically you want to get this as high as possible. The sort of values they're getting with various concentrations of Polysorbate 80 were pretty acceptable, although it must admit it, some of them did take quite some time to get down to sort of the target level of 5 in terms of reduction values. But nonetheless, it did show a lot of promise. There's shown a lot of interest in this. And if you come to another case study, where if you actually, it's a very much more detailed study by Farset et al., where they've looked at Polysorbate 80 in conjunction with TNBP, or tetrabutylphosphate. And what you're getting here is very good levels of reduction at a certain concentration of Polysorbate 80. You can actually achieve the sort of levels of reduction of, a 5-fold log reduction that you can easily get equivalent performance to those regarding sort of Triton X100. So just to give you some examples, there's some published data out there just showing how this material can be used. And with that in mind, we are actually doing our own in-house studies looking at a variety of different envelope viruses, because clearly the more different envelope viruses you can look at, the more convincing the data will be. Moving on, we can talk about protein A affinity resin chromatography. Now, protein really is the primary chromatographic purification step in the manufacture of monoclonal antibodies. It results in a high degree of purity and recovery in a single step. It's really a ligand-based separation, and it's basically used to bind the protein, which can then be washed off with subsequent washes and then basically regenerate the column. Protein A resins are very expensive and they are prone to contamination. So there's always a need within the industry to improve regeneration and also the number of production cycles particular resin can be used before it has to be discarded. And one of the areas that's of interest to us is the potential to use Polysorbate 80 to help improve its shelf life. And it's also worth noting that some of the solutions used to preserve and maintain the columns when not in use, supplied by people like Millipore, do contain a number of materials, some of which could be a role for materials like our super refined benzyl alcohol or our high purity benzyl alcohol, or commonly known as super refined benzyl alcohol, because that, as I say, with low levels of impurities might actually enhance the stability of the resin itself. And there has been some interest within the industry to look at this material in this kind of application. If we go on to the next part of the process, and really the final one, we're now talking about the final fill and finishing step. And this is typically an automated hydraulic sterile process. Surfactants play a key role in this because they are shear protectants and they protect against the loss of the API. And there's actually a publication by some scientists at Johnson & Johnson, or Janssen, as this group's commonly part of, and where they looked at a variety of things like tubing type, operating parameters, and surfactants on particle formation during just peristaltic pump filling and processing of a typical MAB formulation. And the thing about this is it just shows how surfactants can have a key role in preventing API loss, even at the final fill stage, not just during bioprocessing. Typically, if the product's filled into vials along with a final formulation buffer, and you've got stabilizers such as Polysorbate 80, PEGs, both low and high molecular weight PEGs. They can include vehicles such as oils and propylene glycol, and including preservatives such as benzyl alcohol. And the thing to bear in mind here, though, is that there is a commonality both between the bioprocessing and the actual formulations. And it's basically based around Polysorbate 80 and Polysorbate 20, in that they're used in both for very often the same reasons. If you look at the issues, is that basically Polysorbates stabilize proteins and protein-based materials against interfacial stresses. And these stresses can observe during different stages of drug product manufacture. They can happen at the air-liquid interfaces just through shaking. They can happen at surface interactions, as with the processing equipment, where they can stick and bind and clog. They can also happen at ice-liquid interfaces, such as during lyophilization. All of these stresses can cause surface absorption, protein aggregation, and precipitation. In a typical formulation, the concentrations of Polysorbates 20 and 80 are actually very low indeed. They're only about 0.001 to 0.1% weight to volume. Clearly, they're used at slightly higher concentrations during bioprocessing, or can be used at slightly higher concentrations in bioprocessing. But in the final formulation, you need very little indeed to have an effect in preventing what could be extremely expensive loss of very valuable API. Just to give you an example of sort of protein aggregation testing we've done ourselves, this is with bovine IgG with a guanidine hydrochloride, where essentially we're seeing by increasing what's the maximum amount of guanidine hydrochloride we can add before we got precipitation. We can see here we've got a range of different polysorbates of different grades. And the thing of the take home here is that these are all very effective at preventing aggregation. However, there are some other polysorbates out there which are made by a different process called the reverse process, which you have to be aware of, which are not very effective at preventing aggregation at all. We can talk a little bit about those further on in the talk, but essentially, there are products out there that are not made by the conventional process, and as such, they do not perform as well. Which brings me to a point here, which is selecting the appropriate polysorbate for your process and for your formulations can have a critical effect on the final performance of your material that you're producing. So we talked a little bit about bioprocessing and API manufacture and purification. Now I want to talk a little bit about formulations. Typically in formulations, there are essentially two polysorbates that are commonly used. These are Polysorbate 20 and 80. And in formulations, we commonly find both the high purity and super refined grades are used in formulations. And commonly now within industry, you tend to find during bioprocessing, people use the HP grades. And in the final formulations, they use the HP and the super refined grades, depending on the manufacturer. Both of these polysorbates are approved by the FDA IID list for injectables. They have a long track record of safe use. There are other products sometimes used, such as Poloxamine 188, and there is some interest in alternative excipients, but these two materials are still by far the industry standard, and they're used in a wide range of vaccines and monoclonal antibody-based formulations, both with and without adjuvants in some cases. They, however, they are very complex materials. And, sorry, excuse me. Just getting a drink of water. Polysorbates are highly complex. They start off by being, you dehydrate sorbitol. That results in three sugars, essentially. You've got the sorbitol, sorbitan, and then if you dehydrate it further, isosorbide. You then esterify them, and that results with mono, di, tri, and tetraesters being formed, as well as non-esterified products. They're all present. These esterification agents are typically plant-derived fatty acids. The main difference between polysorbate 20 and 80, by the way, is that polysorbate 20 is derived usually made with coconut oil. So its main fatty acid is lauric acid, sorry. Whereas the Polysorbate 80 is palm oil, and it normally contains the highest fatty acid in that is oleic acid. Following the esterification, the material is ethoxylated, and that's a Gaussian distribution with each sorbitol species having a different mean EO mole content. So, for example, with sorbitol, you can get up to 6 points of attachment for ethylene oxide, sorbitan 4, and isosorbide 2. Just to bear in mind another level of complication, because I mentioned the fatty acids being plant-derived. These are obviously complex mixtures, and they're typically a mixture with 3 to 5 different chain lengths at prominent levels. As I say, it's lauric acid for Polysorbate 20 and oleic acid for Polysorbate 80. So here's the standard process. We have simultaneous dehydration and esterification with the sorbitol and the fatty acid to produce both sorbitan and isosorbide. So there's a sorbitan being esterified, and then reacted with ethylene oxide to produce the final product. And that's the idealized structure. Everybody sees that and thinks that's what the structure of polysorbates is based around. And it is, but it's a lot more complex than that, because there are all these other variants creep in, both in terms of fatty acid chain length, the ethylene oxide chain length, and the type of the core sugar itself. Polysorbates, there's a number of them out there. There's huge differences, though, in their purity. There are color differences as one obvious one, but there are also process differences, because with the higher grade materials, you use higher purity starting materials, both in terms of the fatty acid sources and the sorbitol. They're manufactured under milder conditions, so you get less caramelization and therefore less color. And in addition, there's certain grades, such as the super refined grade, which actually uses an additional proprietary purification step. And with these higher grade materials, that means the residual impurities, such as peroxides, aldehydes, and catalysts, are all removed by this additional super refined process. And that's unique to super refined materials. So just to give an example, a tween 80 HP might contain 300 ppm residual sodium, with the super refined Polysorbate 80 will contain less than 5. And we actually have a comparison chart showing the various grades. And the ones who really should be focusing on are those labeled HP and super refined. And that's because these are made to a higher standard of purity, which is very, very important, both for bioprocessing and also for the final formulation. We do not recommend using the standard grades for either application, because really they do contain impurities. And if you have any technical questions, I mean, this chart's going to be readily available. And this presentation is being recorded. Feel free to refer to it. If you have any particular questions, please ask. But essentially, the thing to draw your attention to is things like the low levels of peroxides as part of the specifications, with the additional specification for formaldehyde levels in the super refined grades, the fact that both of these materials are stored under nitrogen, and that we have things like, certainly in the super refined grades, we have the catalyst acid residues removed, whereas they are still present in the HP grade. We do make this material to a very high degree of reproducibility. We have developed our own in-house methods for characterizing these materials down to the various compound classes. And just so to show, this is 6 batches of HP and super refined Polysorbate 80. And you can see we're pretty reproducible across the whole chemical range of compounds in this material. There are some concerns in the biopharma industry generally. One of them is around autooxidation of the surfactant, and this can be due to oxygen, light, heat, and transition metals, often in combination with one another. Hydrolysis. We start off with a polysorbate. It can be esterified. Sorry, It can be hydrolyzed, I apologize, hydrolyzed by either acid-based or enzymatic processes. And essentially what happens, and it's been associated with the reported appearance of precipitate in some formulations. It's actually believed to be down to actually not only the hydrolysis in polysorbate 20, but the liberation of high insoluble molecular weight fatty acids. So here we're talking about fatty acids like myristate, palmitate, and stearate. It is still a major concern in the biopharma industry, and there are a number of ongoing projects, and we have a number of them ourselves with biopharma customers into the various routes of degradation. And we're going to show you some of the results we've been getting up till now. So if you just go back to oxidation, I mean, there's methionine oxidation and there's tryptophan oxidation. Their established routes of oxidation are well known. Methionine, you first of all get methionine sulfoxide being formed, and then the methionine sulfone. And the one to watch out for is the methionine sulfone, because that's an irreversible reaction. Clearly, if you've got these kind of residual groups on the outside of your biomolecule, then you're going to have some oxidation, or you could have oxidation, which could result in a loss of performance of the API itself. Similarly with tryptophan, here the degradation route is again well known. It's a lot more complex, but again, same sort of issue. You can get oxidation of these sort of amino acids, and then you have got a problem. But you can mitigate against it. And I mean, there's obviously various materials you can add, and there's a number of publications talking about the addition of things like chelating agents to mop up residual catalysts or metals, and or the addition of scrubbing agents such as methionine itself to actually just mop up any sort of peroxides being formed. However, one of the things that we offer is super refined materials, and they help in two ways. One of them is regarding oxidation. If you go back to the bottom of the slide, we've got low levels of peroxides in these materials, which are kept low by nitrogen blanketing. They have low residual catalysts, and they have low transitional metals as well, usually at ppm levels. They also have low levels of free fatty acids. So if your concern alternatively is with hydrolysis, you can actually, using the super refined grades, mitigate against that, because the actual low levels of free fatty acid in the sort of material itself is very low indeed. So if you do get any hydrolysis, it's got a longer onset time. And we'll be talking about that in a couple of slides, because that's quite an important point to bear in mind. Whilst oxidation can easily be dealt with and is not so much of a major issue, enzymic degradation is a major problem, or can be a major problem in the pharmaceutical industry, because esterases carried over from monoclonal antibody manufacture can cause degradation of the polysorbates. This in turn can lead to the appearance of visible fatty acid precipitates, particularly with polysorbate 20. And if that happens, that can lead to both a loss of the polysorbate and also a loss of the API stability as well. So not only do you get the appearance of precipitate, but you can actually get a loss of the API and the viability of the actual formulation itself. We've actually done a number of in-house studies, but there are published examples of this too. We actually used a model lipase study and actually followed the degradation. We've been able to follow the degradation and characterize it using a variety of in-house techniques. We have our own in-house HPLC ELSD method for monitoring the degradation of the actual esters. We have in-house LCMS methods for measuring fatty acid liberation. And then, of course, with fatty acid precipitation, we're essentially resorting to visual observations, although we have been evaluating the various instrumental techniques like for flow cam to actually get a more quantitative measure in terms of what actually is the amount of precipitate being formed during this degradation process. But in this typical sort of chromatogram we've actually been able to identify all the major chemical classes of this product, but this is just a schematic diagram of a chromatogram showing essentially what we're doing here over 24 hours is we're targeting with this particular lipase the higher esters and causing a subsequent increase in the monoester composition relative to the higher esters. So actually enforcing the degradation to take place. And this is typical of a lot of the studies we do, both internally and in conjunction with our customers. Just to give an example here, we can look at the non-alkylated species, which are the species which have got no fatty acid, no ester groups at all attached to them. So here we're looking at like free peg, ethoxylated sorbitan and ethoxylated isosorbide over 24 hours. We can monitor those being formed over this time period. And we're doing these experiments, we're running all 3 tests simultaneously, which is quite handy for this kind of study. So if you look at things like the free fatty acid changes, you think, oh, well, they're breaking down at the same rate. We should expect, irrespective of grade, it's not going to change the rate at which they degrade. But the point to notice here is with the super refined polysorbate, the actual, because you're starting at a lower level of free acid, but after 24 hours, you're not getting the same level as you might otherwise get in, say, a tween 20 HP, for example, or a standard grade. It's, typically certainly 2 to 3 times less. And we've actually backed this up particle formation visual studies, where essentially you can see the standard compendial grade readily forms particles. Both if you've got 1 ppm lipase or even 10 ppm lipase, you readily see it, even at 1 ppm lipase. Pre-super refined material is also prone to forming particles, whereas with the super refined, we're getting only trace amounts, if that, in some cases not detectable at all. This is a typical sort of formulation that we made up using a standard histidine buffer at pH 6.2. And the sort of lipase levels you might find as an impurity, when they talk about this in the industry, they're typically talking around about 1 ppm levels of lipase. So we're using the sort of levels that are appropriate for the industry. Excuse me. I mean, one of the things we've done in conjunction with Roche is we've actually can mitigate against this precipitation formation by using the super refined grades, but you can go one step further by actually developing a product where you can actually customize the fatty acid distribution. And this was a request to actually produce a product with lower levels of stearate, palmitate, and myristate esters. It still had to be monograph compliant and multi-compendial, and it had to have the same functional performance. So that's essentially what we've been able to do. We're able to actually make products that are particularly, suited for particular customer needs and applications. So if you come across anything, please let us know. It actually resulted in this piece of work we did with Roche in a publication we did with them, just to show the fact that, we modified the purification, modified the fatty acid distribution in order to minimize the risk of free fatty acid particle formation. So we've got a reference there if you wish to look it up. Just an additional piece of information to bear in mind is that polysorbates, people have come to us and say, well, our polysorbates degrade in solution. And you think, well, why is that? And yes, they will. I mean, they are essentially ester-based materials, and they will hydrolyze over a period of time. But we've done some recent studies where we've looked at various grades. We've got here NF, that's the standard compendial grade material, HP, super refined, and then a specific product we had for R&D purposes, which is basically a polysorbate 20 made from just pure lauric acid on its own. And the thing to note is that basically, if you look at tween 20 over 15 weeks at 40 degrees C, it drops. So if you do make tween solutions, we do recommend you make them fresh, clearly. But the thing about them is that another reason for not using the NF grade, it does seem to break down far more quickly than the higher purity grades. And we've done this both in terms of the total concentration of the polysorbate using a standard industry method, for measuring total polysorbates in formulations. We've also done it with also the Nonesterified content method, which is basically an HPLC method of looking at the various ester distributions. And as you can see, over time, the Nonesterified content rises considerably, whereas with the HP and SR grades, it stays relatively constant over the same time period. Likewise, the Polysorbate 80, same thing. It's not, the NF grade is not as stable as the HP and the higher grade materials. What does this mean if it does degrade? I mean, you're going to get a loss in performance. And we've got a couple of examples here where you're going to get an increase in micelle size, whereas the sort of various standard grades are fairly stable, but there is some increase in micelle size. If you do take a polysorbate that's been made by the reverse process, it is far less stable, and you do get degradation, and you're going to get loss of performance. And I'll just give you a quick mention about the reverse process. The reverse process is actually, rather than taking the sorbitol and esterifying it and then ethoxylating it, what they do is they take the sorbitol, dehydrate it. They then use a reverse process. They swap the chemistry around. If I can go back to the original presentation, the original slide where I showed the actual reaction sequence. The reverse process is different from this one in that you take the sugar and then you react it with ethylene oxide and then add the fatty acid groups on at the end. That's called the reverse process, but it results in a much more inferior performing product. So I was going to just go on to the final slide. And basically just so I hope you realize just how important surfactants can play a key role in the various bioprocessing stages and in stabilizing final formulations. They can play a key role in bioprocessing, particularly with regards to cell viability and therefore yield. And we've generated our own data of showing how high purity polysorbates and customized products can minimize known polysorbate stability issues in final formulations. We do have a number of projects in both fields into evaluating and developing new materials and new variants of these products, both polysorbates and polyoximers, and other materials too. And we have several of these with customers and also with academic collaborations. And if there's anybody attending this meeting today that's interested in wanting to collaborate with us or talk to us further about surfactants and bioprocessing, or any other aspect of biopharmaceutical formulation, please get in touch. And I'd like to thank you for your attention. And I hope I managed to hit looks like I'm just about to hit the 1:48 time over here. So just about on time. Any questions?
Thank you, Stephen, for your interesting talk. I will give the word to Dr. Octavio Donatiu Ramirez.
Thank you, Stephen, for a very clear and comprehensive presentation. I have some questions about the Poloxamer 188. We used to know it as Pluronic FC68, right? And so this the high molecular weight toxic compounds. This is an amphipathic molecule. Have you identified what's the nature of those compounds, and if people produce now the Poloxamer without these compounds? With this?
No, they haven't been identified unequivocally. They are very small amounts in a very complex mixture, and as far as I'm aware, nobody has published what they actually are. But we know what they are in terms of their nature, in terms of their chromatographic performance, in terms of, yes, they're higher molecular weights, you see them by site exclusion. And yes, you can see them by HPLC because they're a hydrophobic group. But in terms of actually identifying exactly what they are, you're trying to identify a mixture within a mixture. And that's something that and as far as we know, nobody else has fully been able to identify and determine. We know how to get rid of them or eliminate them, but we don't. We have not actually identified.
Nobody is manufacturing the poloxamer without the high molecular weights.
At this moment, the way we came into this was that we were actually approached by a number of customers because they were getting all their material from one supplier, and there was a concern about the availability of that material. And that's how we managed to get into this area, because you can see there's a lot of interest in making this material to a certain standard. And so this is why we've got involved and generated our own equivalent material.
I see. And then a second question, in terms of, you mentioned triton is being phased out.
Yes.
For many of the chromatographic steps we use in different bioprocessing stages, we use triton. What other detergent would you recommend? Because some others we have tested, it seems they also present some problems in terms of regulations due to its like the aromatic nature of the compound.
Yeah, well, have you tried polysorbates? Either alone or in combination with others, because they are approved in the final formulation. So that might help you. Now, they're harder to detect because they haven't got an aromatic ring on them. But have you thought about using those?
We've been exploring other compounds, but
also there are a couple of other products that I know that other people have been looking at in recent years. There's a bridge, which is basically an ethoxylated ether, a low molecular weight bridge, and there's also a product called Croda HSHP, which we can send you information on, which you might want to look at.
Okay, that would be great. Yes,
okay.
If you have your, I think Mabel has your contact info,
yes.
Right? We can contact you back, sure. Stephen. Thanks.
Yes.
Thanks a lot.
Yeah.
Thank you. I give the word to Dr. Gloria Saad. She has a question that she already wrote it on the chat, but I don't know, Doctor, if you want to ask directly the question to Stephen.
Okay. Good afternoon. Thank you very, very much for your nice talk. I understand that enzymatic degradation is a problem with your product, but I was wondering if you have just a chemical way to produce this polysorbitol, or is there any plan to produce it enzymatically? I mean, I'm about the esterification and the ethoxylation.
Well, no, I mean, that's by the nature of the product chemistry, you have to esterify it. It has to be an esterified product. So having said that, there are interests in alternative materials that don't have that ester group in them at all. But they are at this moment not approved by the FDA for use in formulations. So there are a couple of other products that have got the same HLB that are not prone to ester hydrolysis at all. And we are interested in these and other materials related to them as potential alternatives. I think one of the things about the ester hydrolysis issue within polysorbates is that it's not as big an issue. I'm not trivializing it as an issue, but it's one that people publish a lot on. But talking to the people again in the industry, it's still very, very widely used. And most times you take the appropriate steps, it's not a problem at all. It's just that they have come across instances where it has been a problem. And sometimes it's easier to write a paper about a polysorbate than it is to write about your biological product because of all the secrecy that goes around that. So I'm not trivializing it, but it's not, people sometimes forget that these things are very widely used, have been used for a number of years with a lot of success. So we shouldn't dwell too much on the negatives, but we have to acknowledge that there can be problems. And if there are, what you can do to sort of minimize them. Does that answer your question or?
Yeah.
Okay.
Thank you.
Does anybody has any other question? If anyone has an additional question, you can write to me and I'll give your message to Dr. Stephen.
Okay.
Well, Stephen, thank you very much for your nice presentation and for attending our questions.
Thank you.
And we'll keep in touch.
Okay.
Thank you.
It's nice to meet you all.
Bye. Bye.
Okay, thank you all.
Bye-bye.
I will type our contacts in the chat box. So if you want to request samples or further information for myself or Steve, please feel free to do that. So thank you very much.